Hello, welcome to Zoominario. Today we're very excited to have Lluvia Flores Renteria. Lluvia is from Mexico City. She got her master's and PhD from UNAM, experimental sciences for her master's and ecology and evolution for her PhD. And she then did two postdocs, one in Northern Arizona University in the US, and the second at the Western Sydney University, Australia, studying the response of pines and eucalypts to climate change, respectively. She's an associate professor in the Department of Biology at San Diego State University. Thank you so much, Lluvia, for agreeing to give this talk. And then please feel free to get started.
Okay, fantastic. Thank you so much, Claudia. I'm very excited to participate in this Zoominario, which is very interesting. And I was really excited to connect with the audience, a very special audience to me. So the title of my talk is Plants Without Borders, Genetic Connectivity of Ecologically and Culturally Important Plants. And so I want to start by telling you a little bit more of me. So Claudia was telling you that I'm from Mexico. This is Mexico City. It's a super nice, very colorful, vibrant city. I know that Claudia is also from Mexico City, so, or at least studied in Mexico City. I don't remember, Claudia, but yeah, so I used to walk around this place and it was very nice at night. I just love so much Mexico City. And this is my university and the bottom left, it's the UNAM. I went to UNAM here. So my the high school, my bachelor's, master's, and my PhD, I studied there. For my PhD, I studied evolution of reproductive systems in pines. And that led me to become more interested in evolution because I couldn't tell two species apart. So I had to solve that first in order to solve my question about the evolution of the reproductive systems. And then with that in mind, then I was offered a position, postdoctoral position in Northern Arizona University. So I continue studying pines and the response to climate change on these pines. And then I got a position in Australia. So this is a picture of the Hawkesbury Institute for the Environment. So I moved to Australia to study eucalyptus and the foliation of some eucalyptus species that are eaten by silage and cause the foliation. But I was looking at the genomic aspects of that. And then in 2017, I started my position as an assistant professor in San Diego State University. And this is my second year as an associate professor now in San Diego. And I moved there because, well, San Diego is right here, right? So this is California. San Diego is right here at the border. So it's a border town. It's very close to home. It's direct flight from Tijuana to Mexico or San Diego to Mexico City. And then I really wanted to go back closer to home. Australia was far away. It was a beautiful place, beautiful time. I really wanted to be closer, but also I was already familiar with the Southwest of the United States and also with Mexico. So that was one reason. The other reason is that San Diego lives, or it's inside of the California Floristic Province. So we can see the California Floristic Province encompasses Oregon, Southern Oregon, most California, a little bit of Nevada here, and also in Baja California. So this California Floristic Province, it's defined by Mediterranean. It's a Mediterranean climate. It has a Mediterranean climate, and it has it's a hot spot. It has exceptionally high levels of biodiversity, and unfortunately, it has high levels of endangered species too. We have something between 6,000 to 7,000 species that are native to this region, and around 40% of those are considered endemic, which means that we have a high responsibility to protect these species in this area because they live there, nowhere else. And now I would like us to pause and just take the opportunity to admire the diversity that we can find in this region. So if you look at these pictures, you can see the color, the function, right? And other things, the forms of these different species. All of these species are very special and very close to my heart because they are species that we studied in my lab, but they are also very important culturally for many of the tribal communities or the native communities in California and Baja California. So they have different uses. So for example, here we have Quercus agrifolia, an oak. These bellotas, or the acorns, are collected and then they grind the acorns and they process them to make flour and make, I don't know, pizzas, or they make a atole. The shawi is also called shawi. And it's a very important food. And there are many uses from these plants. You can use them for many different things. In this region, it's not only biodiverse, but it's also culturally diverse. And in here we can see, I like to think about this region as a trinational region in which we have the US in one side, Mexico in another side, but we also have the Kumeyaay Nation. The Kumeyaay Nation has been divided by the border, actually, and I learned that when I moved to San Diego. So we have this nation divided by the border. And so we can see here these two groups. So the same group divided, in other words. And in this picture, we can see the border near to San Diego. So this city here is Tijuana. And here we have, the hills and the southern San Diego. So we cannot see the city, but this is part of San Diego. And with this in mind, a friend of mine, Sula van der Plank, a friend and a colleague, decided to create a program to study native plants with binational distribution. So plants that grow in the US, but also in Mexico. And that as a means to engage Hispanic students into plant sciences. And the objective is obviously to increase the retention and graduation of underrepresented students in San Diego State, but in turn make them role models for younger students. So we use this approach, right, the tier mentorship approach, in which we train grad students, they become role models, and they teach and empower younger students, undergrads, and these undergrads later go to younger students and they empower them. And so there is this change that gets created. And each of these students focuses in one plant that distributes binationally, but it's a native plant, but is also used as food source or any other type of, it might be used for basketry, but they also have ecological value. And so one of the questions that our students center, or that are usually in their mind, is how artificial and natural barriers affect species connectivity. This is another picture of my friend Sula van der Plank showing the border and how, making the border can also create some effects. Now, just to understand a little bit more of the connectivity, I would like us to think about a species with the distributions, different populations in these kind of amorph circles, right? These areas in green, all of these represent different populations. So sometimes just imagine maybe a plant, a flower, and a bee that is connecting these plants, these populations, by moving the pollen from this big population to a smaller population, so they are connected. So these arrows mean connectivity, so they are connected. But maybe we have natural barriers such as mountains or oceans or deserts that prevent the connectivity, right? So the pollen cannot be moved from these areas into these other islands. And also, I want you to think now about the size of these populations. So when we have a bigger size, we tend to think that there are more individuals of this species here. And with a higher population size, we tend to think in higher genetic diversity. So there is a positive correlation of the population size and the genetic diversity. And when we have smaller population sizes, you can imagine in this population, we have only 10 individuals. And then now these 10 individuals, they are not gonna be mating with distantly related, but they might be mating with their sisters, their brothers, closely related. So that's what we call like inbreeding. So when the population size is very small, we are gonna have high inbreeding. And the inbreeding can cause some effects, maybe some diseases, some other negative effects. So we like to consider a healthy, genetically healthy population, a large population that has lower inbreeding to avoid certain negative effects. But and on top of the natural barriers, now with humans, we have the artificial barriers, and this can also pose a negative effect in the connectivity. In other words, they can structure the populations. So our students ask these questions. Do we have connectivity? Is this species well connected? What's the genetic diversity in these populations? And so we train them in the field. So obviously we have to take them to the field. We train them in the lab, so they acquire very strong lab skills. And then they have all of this genetic research, this analysis, right? So all of the bioinformatics and also the analytical, statistical approaches for it. And all of that allow them to make decisions. So what are the best practices? So what are the best practices for conservation, for management of this species, but also for our tribal partners or native partners? What are the better sources of food for them to be collecting, for example? We also engage with the community, and with the community, we explain them how the planting native plants instead of having the grass is super important. So by planting native plants, you're going to reduce the amount of water. So this is in the left here, you have the traditional landscape with a bunch of grass, and you need a lot of water. They produce a lot of waste and maintenance. But with native plants, you reduce all of that. So you use less water, which is an issue, especially in California. Right, so we reduce the amount of water, we reduce the amount of waste, and the amount of maintenance. So all of those things help us to increase the engagement of the local communities and to teach people the importance of the use of native plants, the conservation of the native plants, evolutionarily, but also ecologically. It's important for us to connect at the binational region, right? So making efforts, individual efforts is not enough. We need to work as partners between Mexico, the US, but also the native nations need to work collectively to save our species. So that's what I title my talk, Plants Without Borders, because the borders are imposed, right? So we really need to break those borders down, yes, and then help us. Now, I want to show you some of the pictures that we have here for our program. So we take our students and we have immersed our students in tribal communities. In Yauquatai, for example, they are here weaving using juncos to make baskets. We take them to the field, as I was saying. We train them in the field exploring. We make a bunch of experiments. So this is an experiment that Carlos is helping us taking care of these oaks. Then we go again to the field. We train them into the lab. In the lab, PCR, DNA extractions, sequencing, all of those things that we do. And just having fun and creating community and friendships. Now, if I have time, I would like to give you two small little pieces of things that our students have done in a very superficial approach here. So the first example that I want to share with you is this Ensilococcus bicolor. This is a beautiful species here. And this was part of the master's of Kyle Gunther, who graduated 2 years ago. So this is a shrub. So you can see here this shrub, it has multiple stems here. It's very interesting because it's adapted to fire. So all of these here is called boral, and it's kind of like a stem root underground that when when we have a fire, when we have fire, all of these at the bottom, at the top, will burn. And the thing on the ground, this boral will survive and it will resprout again. So it's a survival mechanism. It's adapted to fire. Now we have these flowers here, and you can see the name Xylococcus comes from the fruit. The coccus means spherical. So this is the fruit, it's spherical. And the xylo means woody. So this, with time, it will become very woody, the fruits. And bicolor comes from these, the two colors, and that refers to the leaves. So under the under part of the leaf, we call it the baxial side of the leaf, it's whitish because it has a lot of trichomes, a lot of hairs. And the top surface is green, very, very green, and it doesn't have these hairs. So you cannot appreciate it here, maybe in this leaf, but that's the bicolor, that's what it means. So what we did is that we went and collected in all of these populations and the distribution of these species, Xylococcus bicolor, the Mission manzanita. And here you have Kyle in all of these places. In some places we had to go and have mules, and the locals help us too with the mules, and they join us in some very amazing adventures. So this is a whole distribution of the species, and these are the populations that we collected, that we have material. And these are some of the results that we found. So let's see, let's start from this one. So this is a PCOA, and this is an analysis, it's a multivariate analysis. These are not units, it's a unitless approach, but it helps us to understand the genetic distance. We can understand that. So the closer the dots, each dot represents an individual, and the closer the dots, the more genetically similar the individuals are. And we color these by 3 different colors. So these three colors, these 3 populations here are in the mid peninsula of Baja California. And then everything in green here, well, it's more in California and in the northern Baja California, and the Cedros Islands over here. And this is Catalina Islands. So as you can see, Catalina Island, the individuals from it tend to cluster together, right? All in purple here. Then the green populations also tend to cluster more or less together. And then we have a small gap that separates the populations from the mid-peninsula, okay? So we found like 3 main clusters. And then we did an analysis called a structure that allows us to identify how many genetic clusters do we have in our populations. And the optimal, I'm showing here 3, so with k = 2, k = 3, and k = 4, meaning we have 2 genetic clusters, 3 or 4. The optimal was k = 3. And this is consistent what we found in our PCOA that I showed you earlier, that I explained you just now. So in the left, we have the different populations, the names, and each of these lines, if you're not familiar with these lines, they are individuals, and they have the proportion of the genetic ancestry. So for example, here it says that this individual has almost 100% of this ancestry belongs to the cluster that is in orange, right? And this individual over here in green, it belongs to the genetic ancestry that is represented in the green cluster. I told you that the optimal was k = 3. And you can see that the Catalina Island population, the CI, is the one that we have in purple, also in the map. And then all of the populations that were in green tend to be in one in one cluster. And then all of the ones in the mid-peninsula in orange are here represented in orange. But we can see also that they are admixed, meaning that we have genes that are passing from the cluster in orange that are more northern, and they are admixing. So they have gene flow. And we have here at the bottom some genetic differentiation analysis, so the FST that we call it. And FST goes from 0 to 1. 0 meaning there is no structure among the populations, meaning we have random mating, and it's also going in all possible directions, right? So there is no structure. Whereas 1 is that there is a barrier, maybe, that there is completely isolation between these two populations, right? So these values are relatively low, but we have some type of differentiation. So the smaller differentiation is between the north and the south. That means this green and orange, they have differentiation, but not that much compared to the south versus Catalina Island. So that means the orange and the purple are the most differentiated. So what is it telling us then? So what it's telling this is that we have 3 groups. So all the analysis that we did, and we did more, is that we have 3 groups, but we have certain barriers. So let's see what these barriers are. So here in between the orange and the orange and the green, we have the desert. This is the Cataviña, is right here. And this is the vegetation that we have. So we don't have Xylococcus bicolor, and this was recently formed, actually. So it makes sense that there is still a little bit of the differentiation is kind of small, because this just happened. The formation of this desert is kind of recent. The other barrier that we have is this ocean, right? The Catalina Island separated a while ago, and now we have this separation between the island and the mainland. Now you are probably wondering, what about this island? This is Cedros Island. This is probably something that we need to continue. This is in the next step type of questions, because we don't have that many individuals. So this might be an artifact, and the lack of individuals might be grouping it together with this group here in the main island, in the mainland. Yeah. So what we want to know next, and we're doing some analysis about it, is when this, with the genetic data, we want to estimate the time of divergence between these 2 groups or 3 groups. And that's something that I will present later. Now, the next small project that I want to show you is in Pherocactus getziai. This is a beautiful, beautiful flower of Pherocactus getziai. It's a cactus. And this is led by Jasmine Lommel. She's doing her master's, so she's going to graduate soon, hopefully. And she's doing here some, well, we're doing some experimental crosses that you're going to see in a few minutes. This is the place. So this is amazing for a student to be doing a work in a paradise like this. These are the islands of Bahia de Los Angeles. So this is a natural laboratory for us to assess the genetic connectivity among islands. And the islands differ in sizes, in distance, and they also have different geological histories. So it's very interesting, these islands. So these are the islands. There are around 16 small islands in this region, but only there are 7 that have this cactus. And that is because it is associated to the type of soil. So if they are metamorphic, they can grow there. Otherwise, they won't grow there. So we published that just recently, well, 2 years ago. But now, this is also very important. Studying these species is super important because we only have less than 500 plants total in all the islands. So it's at risk, this species. This is the fruit of this plant. These are the flowers. These are beautiful plants. They are cylindrical with columns, as you can see here. And well, the question was to determine the connectivity within and among the islands. And we have two approaches. One is experimental and the other one is genomic. And this is just an example of the islands, 5 islands that we are focusing for this study. And so for the first approach, which is looking at the connectivity through pollinator dispersal, what we did is that we wanted to check if the pollen can be moved inside the island, but also across the islands. And what we wanted to do is to track the pollen. So how do you track the pollen? Well, you go and dust fluorescent dyes on these flowers. And this is what we are doing here. So we are dusting the pollen. So then later when the bees come visit these flowers, they will take the pollen, but will also take the dye with them. And then at night, we will be going to all of these islands. Well, during the day, we dusted them and we went to different islands and we color different islands with different dye. And at night, we could see what colors they were fluorescent. So this is a plant that we didn't mark, but then it had dye there. I don't know if you guys can see it. It's very, very mild, the intensity, but it's green. So this bee collected the pollen with the fluorescent dyes and deposit the pollen here in the stigma and the female part of this flower. And that's how we know the pollen is being moved inside of the islands, but we wanted to check if it was being moved among the, among different islands. So what we found, like for example, La Pata, this is the name of the island is La Pata, here represented in blue. What we found is that we targeted 36 plants. So we only dyed a few of them, right? Like 5, I don't remember how many now. But we imagine we have just dyed 5, and then we surveyed 36. And we found that in 22 of these, we found dyed, meaning that the bees visited these 5 plants and dispersed the pollen to these 25 plants. Then we have other 14 that they didn't have dye. So that means that by chance, the bees didn't visit these plants, these 14 plants, okay? So the same thing happened in La Bota. La Bota is represented here in purple, and in there we surveyed 25 plants on top of the 5 that we dyed, and 19 had dyed, and 6 of them didn't have any type of dye. So this is represented by the gray. Then it starts getting more interesting because for La Cerraja, we surveyed 71 samples, 71 plants. 34 had the dye, and 36 didn't have anything. But then we have 1 that had the green and the blue dye. So meaning that a bee brought dye from here all the way to La Cerraja, right? And then, so we have tiny little bit of gene flow from La Pata to La Cerraja. Now in La Ventana, which is a bigger island, we target 78 samples. 43 had dyed from La Ventana, so orange dyed. And then we have 19 that didn't have any dye. But then suddenly we have 12 plants that have gene flow from La Cerraja. And it kind of makes sense because it's nearby. And also we have 1 plant that had orange, green, and blue. So 3 plants that have orange, green, and blue. And then we have 1 that had orange and blue. So again, moving the pollen from very distant to, distantly. So that was our experiment in reality, right? So the connectivity. So we saw that there was a lot of movement of pollen inside the islands with a little bit of pollen movement with islands that are distant. And the closer these islands are, the higher the gene flow. Well, we didn't find that for La Pata and La Bota, and it might be, I don't know how to explain that at this point. Then for the genetic approach that we did, we could see here that if we color, this is similar to what we explained earlier, the PCOA, where we have the closer the points, the more similar they are. So we have around 10 individuals in each of these islands. In the case of La Llave, for example, because it's a very smaller island that you didn't see earlier because we didn't include it in the experiment, and I'll tell you why in the question session. La Llave had only around 7 individuals total in the whole island. So we included all of the individuals that are in the island, except for 1 or 2 that were very small. We didn't want to injure them. But again, we see this kind of different clustering. La Pata over here, La Bota, La Flecha, La Ventana. So they are somehow separated, right? But this is based on how we color these points. When we did the structure analysis that I explained you earlier, that's similar to what we had. So each of these lines represent an individual. So you cannot see, but in this block, we have 2 or 3 individuals. So it's hard to separate the lines, but each of them will represent 1 individual. So in all of these call rate 60, 70, 80, and 90, we have the optimal grouping or clustering is 2. Now, I want to talk to you about the call rate. This is how many SNP we are including. So how many genetic markers we're including. If we allow for missing data, if we allow for a lot of missing data, we have 60. For less missing data, 70, and so on until we don't have any missing data, right? So, but we have thousands and thousands of genetic markers still. Okay, so now, for most of these analyses, regardless of the call rate or the missing data, the optimal is K equals 2. And this is slightly different to it's not representing each island, right? If we have every different island representing, the optimal K will be 5, right? Or 6 here, sorry. But we have K equals 2, and that is because somehow these 2 islands are more connected, and these 3 islands are more connected, and La Flecha, which is in between, is over here in this region, over here, in which we have admixture, but also we have individuals that look more like the southern region over here genetically, more towards the south, and others that are similar to the northern part. So somehow we have a differentiation that splits the northern islands versus the southern islands. But of course, if we reduce the amount of SNPs, we lose that signal, and it doesn't have any differentiation with call rate of 100. So I just want to, because I know that this is kind of very, very fast, and wanted to close by saying that this program has been very successful at training our students, and they are approaching these questions to understand how our native plants are doing and how they evolve in the past. And I'm very happy to be here. So I just want to thank about a lot of my students, especially Kyle Gunther and Jasmine Lommel, who I used their results for this presentation. But definitely, I have been honored by working with Sola van der Planck, the binationalist species group, I'm sorry, and the Resilient Restoration team. And a lot of people have contributed to this program in both sides of the border. And definitely all my students that I'm missing a lot of here, and the funding sources, we have received a lot of support from the USDA and NIFA agencies, but also NSF and many others. So then I was told that we could jump into this now. Claudia, I think is appropriate.
Yes.
Okay, so I was thinking like, okay, I was asked to share a little bit of myself. So, and because it's Dia de Muertos, it was Dia de Muertos recently. I dress as Katrina to honor my ancestors, but also to join my little niece and nephew to ask for the calaverita. So when I was little, it was a very important day. So Halloween is different, totally different from Dia de los Muertos, but in Mexico, we also ask for the calaverita, which is kind of similar to the trick or treat that we have in the US. And then we were just wandering around, but we make a big deal with the offerings. So we can see here some of the offerings. And this is CEU, UNAM. This is the library, a beautiful building. And every year I used to go there and tour around the offerings for Dia de los Muertos. And so people dressed up, and then they put the offering for, you know, to have the memories, to honor the deads, our relatives, right, who has passed away. And it has been a lot of fun. So this year I dressed up like that, and I want to share with you, and I hope you like it. And I don't know if you guys have any questions.